I got it here. Okay, I'm going to, or I was asked if I could just recap a bit on the MRCs and the effect that it has in prudent use of antimicrobials, or in general on antimicrobials. It's a topic which is quite close at heart. And I think what I did here, something that we as veterinarians do particularly brand badly, we don't brand ourselves. And that's the reason why I branded the we and us and ours and put the trademark sign on. And who are we? That's everybody sitting in here, the poultry vets, or we should actually go a little bit wider, and it's actually SAVA. And I think we should make a much bigger effort to brand what we are doing so that the public is more aware who veterinarians are and what the veterinarians actually have for an impact. And if we would do that, I believe we could have a bigger say in how to use, now in this particular case, antimicrobials, but in general what the veterinarians are actually do for the public. So what do we talk about? If we are sitting here and we talk about the impact of MRCs on prudent use of antimicrobials, we obviously think of a poultry or a broiler house, but it goes much further. I think we must think in general, what does antimicrobials actually do to us? And to us here means the whole population of South Africa, where we are busy, where we're living, where I think we all want to make a contribution to the well-being of everyone. So it's again the trademark of South African veterinarians, we will really start to think a little bit wider and not only think and do the right thing, but we must also consider how can we do it so that the general population, the country, and in particular the politicians, do actually notice what we are doing. So whose interest do we represent? We don't actually work for large corporations or for private companies. We actually work for the South African population. And I think if we get this mindset right, then possibly we are starting to do a few things slightly different. So our goals should always be, or our attempt, that we are relevant where we are living, and that we are relevant now, and we hopefully stay relevant in future. We often talk of food safety, and food safety is to make sure that we produce animal proteins which come on the table of the South African population, which are healthy, which Osal told us how we must, or how to check for residues. And obviously that's important, but apart from the food safety, we also have to ensure food security. And I would like to bring this a little bit in perspective. I'm sure we are all aware of it. But at the moment, we are about 60 million South Africans. And if you look how we are going to increase, or how the population is going to increase, we need to produce more animal protein. And I think animal protein is very important for poorer communities, even if they can barely afford it. We have to make sure we will put protein on the table that everybody in the country can eat. I found this very interesting graph. So it's a ratio of the extensively grown animals or livestock compared to the GDP. And if you look here at the blue point, if we have a GDP of $2.9 and we increase this to $3.8, so we actually reduce the extensively grown livestock from 80% down to 20%. What does this mean? We grow in population size, and proportionally or inverse proportionally, we will grow the extensive livestock production. Obviously, because people have a little bit more money, they can afford to buy a chicken. They don't all need to grow it themselves. It's a trend to go into urban areas, and it all culminates in the same thing, that we actually grow together and we have to produce this food, and we must make sure it's available and it's safe. And we must do this. So if you look at the population globally, just to give a little bit of an outlook, I was quite impressed in the projected changes. So Africa is going to grow tremendously. It's now not just South Africa, that's Africa, but it's the same general picture like I showed in the growth curve. So we must really make an effort here in South Africa that we are able to produce the food that we need for our community. And if Europe is trying to change this and having some of the rules in the Western world or in Europe and America, which go a bit overboard, I think we must, they don't go overboard. I think it's right if you can afford it. But the first step for us must be to make sure that we can provide the food that our population needs, and we have to make sure that we do this locally and we do it now and we do it for our people. So what's our problem with antimicrobial resistance? That's a typical picture of a human being which was unfortunate enough to be infected with a multi-resistance Staphylococcus aureus, which is resistant to third-generation cephalosporins. You can hardly treat it, and there are currently thousands of people dying from it. And I think this very drastic increase was actually one of the initial triggers that this antimicrobial resistance became such a topic, not only here in South Africa, but obviously globally, and both in the human sphere as in the veterinary area. Okay. So if you want to test for antimicrobial resistance, you can do this in two ways. We can do an antibiogram, or we can do a minimum inhibitory concentrations. And for me, they are fundamentally different. So I'm going to go in some details to it. That's what I was asked to show today, but I thought I'll try to put it a bit in perspective. So the swap, when we started at VTech to do these minimal inhibitory concentration profiles, we received many swaps which had multiple cultures. There was a total chaos, and then we realized that a lot of the surveillance swabs, they were taken from the trachea of birds. And so a swab from the trachea of a bird, I think if you do what Jaco did, wonderful. If you want to survey what bacteria are present, that may be a good idea. But if you want to define what's a potential problem in a specific flock, it's absolutely useless. So one cannot take endotracheal samples. It has to be from a pathological lesion, which we usually say you must be able to see it macroscopically. And if you do a culture, then almost invariably on a good swab, you have a single culture. Almost all bacteria, they behave like a conservative village. They like whatever is there, so they like their own, and everything foreign, they do everything to kill. So if bacteria grow, they will produce substances which prevent any other bacteria from growing. So we need to have a pathological lesion. We need to culture it, and once it's cultured, we need to confirm that what we see are actually E. coli or Salmonella or whatever we think that we are dealing with. We now do all the work together with SMT. It's phenomenal to work together. Thank you. I think one can see that it's a lab run by veterinarians for veterinarians, because it's just a different approach to how we do things together with SMT. I'm allowed to do marketing here. I said a bit in the beginning, we must do more marketing, so I'm doing that. So with an antibiogram, we do these disc diffusion tests. We have different antibiotics on different discs. We put them on an agar plate. We look what the inhibition zone is, and the larger the inhibition zone, the more sensitive, the more resistant the bacterium is against this specific antibiotic. The problem that I have with this, once we have an inhibition zone, we open a table and we look at the table if it's resistant, intermediate, or sensitive. But what does this really mean? And if you go back and you actually look in literature where the data is coming from, for different antibiotics and for different inhibition zones, that's not an inhibition zone which is from a poultry, but some of them extreme is for fosfomycin, that's from human bladder infections. So we work from a human bladder infection on an inhibition zone, and we try to make a conclusion on a broiler house. I think that is very far away from where I think we should go. If we do minimal inhibitory concentrations, we have a row of different dilutions. Let me just see. Can you help me here quickly? Doesn't do anything. Oh, that's fine. Okay, so we put in all the different tubes, we put bacterial broth with bacteria, the same number of bacteria, and then we take a stock solution which has twice the concentration of a specific antibiotic that we want to have in the highest concentration. And if we have in these tubes, for example, in the microplates, usually 100 microliter, then you add 100 microliter of this concentrated stock solution, and you then end up with, in this case, the 32 micrograms per milliliter. And then you mix it well, you take half of it out, you put it in the next tube, and so you go with your line all the way down, and the last one you take again, half of it out, and you discard it. So now we have a decreasing concentration, always halved, of the antibiotic that we want to test. That's how it's done in microteter plates. We have 12 rows from left to right, usually row 12 from top to bottom. That is our control, where we have no antibiotics. So in every well, that bacteria should grow nicely. It's a bacterium where we know also later what the resistance should be in different controls, but it's a bacterium that we know should grow in this broth. And if everything goes right, then obviously we have growth. In the other ones, we have from the bottom to the top, a decreasing concentration of the antibiotic. So in row H, we would have the highest concentration, and then going up to row A, we keep on halving the concentration. And depending on the antibiotic that we are testing, we start with different highest concentrations. And if we look at these plates once they are finished, and we look on it and we just see a clear fluid and nothing on the bottom, we then put the plates for about 18 hours at 35 or 37 degrees into an incubator. And in this time, in the positive control, the second from the left, there should definitely be bacteria growing, and then it grows up to the level where it can until the antibiotic concentration is actually becoming too high, which would then be our minimum inhibitory concentration. If we look at this from the top, we would see a sediment at the bottom of the tube. And if you go at the same plate that I've shown before, after incubation, you can see in the row all the way on the right, you can see these tiny little dots. I magnified this a little bit. So in row 12, all the way on the right, you see a white dot, which is the sediment of the bacteria which were growing at the bottom of the well. Can you see that? And in row 11, you would see that the second last still has a little bit of growth, and from there on, it's a totally clear well. So the minimal inhibitory concentration would then be the third from the top, which is C. You go to your table and you look what it is. So it's straightforward if everything goes right, but it's quite complicated to get it right. Right, then we started to put this into an MIC profile where every column is a specific antibiotic that was tested, and every row is one bacterial strain that was tested for all the different antibiotics. And over the years, in the beginning, we didn't really know how to code this, but then we did a lot of work in the beginning with serum levels that we could obtain, and we looked clinically where we had success with treatment. And I don't like to talk of resistant and sensitive. I like to call this treatable and not treatable. And treatable means with a reasonable amount of money, we can give enough of a specific antibiotic so that we have a success to treat or to control a specific pathogenic E. coli in the birds. The blue ones would be our breakpoint or critical line where it's getting too expensive and where we can't actually reach the levels. Now, the reason why I don't like to use the published breakpoints for fosfomycin, again, as an example, the published breakpoint would be, if I'm correct, it's 128 micrograms per milliliter. And if you would want to achieve about 64 or 32, which would what we need to get more or less to a level where we have an effect on the bacterium growing, and somebody would come to you and say it costs probably about 25,000 rand a ton of feed to put the medication in, then you would most likely not want to talk to me anymore. So that's why I like to call it treatable and not treatable rather than resistant and sensitive. Now, the really nice thing about MICs compared to an antibiogram, if you have an MIC level of 32 or 16 or whatever it is, that's directly the concentration that we measure of the antibiotic in the blood. So if we bleed some birds and we measure the serum levels, we can directly relate the serum level from the bird to the MIC level. And in human pharmacology, we aim to have 4 times the MIC level in the blood compared to the MIC level, which inhibits the bacterium from growing for a successful therapy. In birds, we do some herd health, and this level can be much lower. And we are actually, if we are half or just a bit below of the MIC level from clinical experience, we do well, and we actually control the infection in a specific flock. So probably the sick birds, they won't really be controlled, the really bad ones, but the majority of them, which is not so bad, we just support their system, and the birds, and that's enough to help them with their own immune defense to actually get over the infection. For many years, to my big frustration, the Department of Agriculture didn't want to know of all our MIC data, and over the years we collected, I think we are currently at about 260,000 MIC points, and the Department of Agriculture said it's useless data because it's a private company doing it, it's a compounding company do it, we don't know how you do it, and it's useless. When the OIE started to ask for information on reports from a surveillance in South Africa, we had some meetings with Moritz, and he was actually quite excited about our data. And the last couple of years, we give this to the Department of Agriculture every year to report to the OIE. And there are some drawbacks because the resistance data should actually be spread all across South Africa through all the poultry which is there, would go from backyard chicken to wild birds to intensive production. We do not have that. So we specify that it's from intensive poultry production in South Africa, but I think seeing that this is the majority of the protein that's eaten, it's still very valid data. So let's look a bit what's causing antimicrobial resistance. So bacteria are genetically trained to become resistant. If a bacterium wouldn't become resistant and it's growing and next to it is another bacterium or there is a fungus or something, they all produce some antibacterial substances and they would long have died. And if you think that we probably originate originally from bacteria or from even minor organisms, they went a long way and they had to find their defense mechanisms. So the one thing we know, bacteria, they do develop resistance. We can do whatever we want, they do. If they are exposed to it, they will develop resistance. For many years, one thought bacteria would never develop resistance against colistin because there was a totally different mechanism. And we know the bacteria have proven us wrong after about 50 years of intensive colistin usage globally in the livestock industry. What is also very special is that genes are actually transferred. How genes are transferred between from one bacterium to the next. So if you look at the anatomy of the bacterium, we have plasmids, which is a circular piece of DNA, and on a plasmid we can have a few hundred bacterial resistance genes, which is causing a huge problem. Not resistance genes, a few hundred genes, and we can have many different resistance genes on one plasmid. So if we look at how bacteria can transfer genes from one bacterium to another, there are 4 different mechanisms: division, conjugation, transformation, and transduction. I'll quickly go through these. If a bacterium divides, we split the bacterial DNA, we split the cytoplasm, and basically we have twice the same that we had before. In case of conjugation, it's kind of a French kiss. So bacteria, they form a bond between the two bacteria, and they exchange some cytoplasmic material, and plasmids go from one to the other and from the other to the one. So if there are plasmids with resistance genes in one bacterium, they were just transmitted to the other one. For transformation, if a bacterium dies for whatever reason and is falling to pieces, the next one comes on the boardwalk and sees some pieces of food lying on the ground, just licking them up. And instead of digesting it, it's actually integrating the DNA particles into plasmids or into the DNA of the bacterium. Especially this is a huge problem. And then transduction is where genetic material is transferred by bacteriophages. So we did a lot of work with the University of Bern, where we wanted to evaluate if there are specific resistance patterns and if we can do a specific resistance profile for a bacterial population in an operation. Okay, I'll go a little bit fast. So the results were shocking. So we saw that only 9.5% of the bacteria had no resistance. And from up to column 2, there are about 30% of the bacteria which had less than 3 resistances, and we had some bacteria which had 10 resistances in one bacterium against 10 different antibiotics, an absolute disaster. That just shows the different antibiotic classes in the blue bars and the resistance level. Then we also have the 3rd generation cephalosporin resistances, which are also amazingly high. It's about a quarter of the bacteria which had 3rd generation cephalosporin resistances. If you only look at these, which are absolutely essential, if you look at the interaction from what we are doing to the human population, obviously against the 3rd generation cephalosporins, there was 100% resistance, but the same bug had also incredibly high resistances against many other bacteria, like the extreme ones, tetracyclines and sulfonamides. We then did a genome sequencing of them, and we were hoping, like I said in the beginning, or the goal was actually to see some gene sequences, and we hoped that we could formulate, based on this, some strategies for different operations and how to treat, and we saw we could not do that at all, because they were basically all of them totally different. Now, after Jaakko's very interesting talk, I really would like to get a copy of his thesis. We also did a whole genome sequencing of, I think, a bit more than 200 bacteria in total. And I selected now quickly few which had no antimicrobial resistance. But if you look at the virulence factors measured with 2 different methods on the right, you see that these are totally different for individual different bacteria, which have no antimicrobial resistance at all. So the two, they really have nothing to do with each other. Okay, so what does this mean for us? So antimicrobial resistance is a fact. We have a high diversity. We must be actively involved in surveillance programs whenever we use antimicrobials. I think we shouldn't use it without establishing a MIC profile so that we know in advance what we are going to use if we have to. If there is an outbreak, obviously we should still take a current sample to confirm that our profile is more or less predicting what we now have. And I think it's very important that we engage with authorities that we can share the information. So a practical conclusion And we saw this in our MRC profile. So because on one plasmid, it's possible that we have a multitude of resistance genes. It may be that we treat a flock of chicken with a tetracycline, and we see slowly that the resistance against fluorphenicol, enrofloxacin, ampicillin is rising as the resistance is increasing against the product that we are treating with. And the reason for this is because the bacteria or the resistance genes may be on the same plasmid. So we don't never have to use another antimicrobial, and we can still push up this resistance, which was quite amazing for me to see this first in theory. And then we had operations where we could really see this. And we actually sent some samples at the University of Bern, and we could confirm it. Okay, so for the few treatment options, we must really take care. It's absolutely essential that we are specific. If we talk of specific, that means we must look for a specific farm or an operation. We must look at the time now when we are doing it. We must confirm we have the right bacterium. We must know the MRC level, and we must know how much of medication that we need, or what feed or water inclusion that we need to get to the right level. We must combine this, and that's absolutely essential, with the clinical findings from the veterinarian. And we must be certain that we are actually dealing with a bacterial infection and not, for example, with a metabolic issue. We must be effective. Again, when we started and we didn't really have the knowledge that we gained later, but we realized soon that feed mills often had enormous variation. So to be effective, I think we should measure at least sporadically the inclusion levels in feed mills, which I think today is standard. When we started with this 15 years ago, it was never done. And then we should also confirm serum levels so that we know that with a specific level of medication, what serum levels we have, so that we have a bit of a trace to our MRC. And then it must be safe, obviously. So we must somehow have withdrawal data, which we can either do through trials, we can read in literature, and we should, on top of it, do residue monitoring to be sure that we have a safe product for the consumers. So it's important that we have a diagnosis, which must be done by somebody sitting in this room. It must be the veterinarian, which has a clinical finding, working together with a lab where the MRC is done. And then if we have a choice, we should use the published literature so that we can actually use the less critically important for humans first, and only if these don't work, then we can go higher up in the list. So what Europe is currently doing, they have a target of using less antimicrobials than 50 milligrams per kilogram of meat produced. Some of you have had operations or cycles where the total usage was below, I think, 11 milligrams per kilogram, which is phenomenal. But if you look at the data, we are, I think, globally still far away from the goal, but it can be done, and I think that must be the goal. In America, one obviously shared the same concerns of the WHO, the WAH, or the EMA, is that antimicrobials were used, especially in the States, the same way as here as our stock remedy, where one could buy antimicrobials over the counter. America has then introduced a veterinary feed directive to address the main concern, so that feed can only be medicated on the prescription of a veterinarian. And I think that's really would be an important step for us to do, that we don't have stock remedies where you can buy antimicrobials and put it in tons into the feed without the vet being involved. I think that's really wrong. So we need to remain engaged. I think it's very important that we push our brand as veterinarians, as livestock veterinarians, together with SAVA. It mustn't only be poultry, so that we are known in the industry. We must be known by the general population. So if there is a problem somewhere with a residue, they should actually first run to veterinarians and say, give us our opinion. Give us your opinion. So they shouldn't first go to everybody else, and in the end we read of it in the newspaper, and then we are running to try to still give a comment. I think if we could change this, we would have made a big difference. And I see this as very important. Because if you want to retain the antimicrobials that we currently have, which we have to use if things really go wrong, I think we must show that we are the leaders. I think we must show that we have the interest of the public at heart. And we must be seen as the guys which are actually supporting and promoting food security and food safety. Thank you.
Thank you, David. Thanks for the interesting talk.
Thank you.
This is for you.
Thank you.
Any questions from the floor?
David, so obviously you said that it can persist in plasmids, the resistance, even for a product that you're not using. But for something that colistin that we know is not used, have you seen a major shift? Because I just saw that one slide that looked like a 0%. Could you see a major shift over the years?
I thought somebody might ask this. In our MRC profile that we do every year, we usually plotted this, since we do it I think we started in 2007 or 8. And colistin, we always saw a resistance, which was typically somewhere between 2 and 4%. In one year, we had a bit of an increase. Then in 2014 and 15, we suddenly saw a massive increase in colistin resistance. We were already working together with the University of Bern. We immediately phoned my friend there and said, hey, there is something totally wrong here. I don't know what it is. And then in November 2015, there was a Chinese publication where they detected for the first time this MCR1 gene, which is showing the colistin resistance. We immediately sequenced it, and we could, before the end of the year, confirm that it was the MCR1 gene that we saw in South Africa. And I think on 11 January 2016, we sent a letter to all the poultry vets and said, hey, there is a huge issue with colistin. We must stop using it. And we have never used it since. About 9 months later, the government banned colistin from using it and made it a Schedule 6 substance, which I think was a very wrong decision, because there are cases where one should still use it, but one should use it very controlled. Now, with colistin specifically, because it was a new gene emerging, we saw here locally, if you look at the resistance level where it went, and today we are again sitting in the region of 2% resistances, although we never use it, but it immediately came down. So obviously it's not the same as the others sitting on the same plasmid and widely distributed. And the same was observed in Holland from tourists which went to Thailand when they continuously monitored the MCR1 gene in the feces of the people coming back with colistin resistance, but over time very soon it dwindled away. So we were very lucky there and probably fast enough to react before it was pushed. So at the moment, I think South Africa is the only country where it's not forbidden, but where it's a Schedule 6 substance.
Okay, thank you, David. There's another question.
Thank you. My name is Wesley. I would like to know, I get the impression that you prefer the use or make reference to MIC data as opposed to antibiogram. Is that correct? Yes. For recommending what antibiotics to use on a farm.
I very much favor MRCs over antibiogram because it gives you a direct translation of serum levels that you should get. That's
all right. So my next question is, in the absence of this empirical data, say I'm thinking at the back of my mind that it may not be perhaps the same from one farm operation to the next, depending on how they've been using antibiotics in the past, or probably some other compounding factors along that line. Would you still recommend that a person goes with antibiogram in the absence of this information? because there are instances that I've asked the lab to give me data like this, and they say they cannot give me, they can only do antibiogram. So what next?
Absolutely. If you have no facility to do it, you should use antibiograms. It's certainly better. And if you have nothing, then you should probably alternate antibiotics. For me, I think we also changed the treatment strategy. Today of the opinion, if you do continuous MRC monitoring, you should never ever change the antibiotic before you see the resistance going up, because you should rather use one, and as long as you keep monitoring, you know it's resistant. It's not resistant yet. There is no reason to change. So what we did in the past, where you alternate antibiotics, I actually think it's bad because you have more of a chance to select something which is resistant. If you have nothing, you must use antibiograms. And if you don't have antibiograms, you must just change. And what I also think in terms of treatment, what we always try to do is that the entire vertical integration from parents, grandparents, broilers, if possible, are put on the same treatment if the MRC allows, so that we use for everything the same product until we see in the change in a resistance profile, which you are now monitoring, what one never did in the past. And only then if you change, but then you change everyone.
Okay, thank you, David. Any more questions? Okay, thank you.